This is part 5 of 5 on chapter 9, Microbial Genetics. This part is looking at how bacteria can increase their genetic variability. So remember, bacteria, they divide by a process called binary fission. And it's basically bacteria cells just make clones of themselves. So if you have one bacteria cell that's just making clones, all of those clones are genetically identical to each other, which isn't good. If you're all genetically identical, it just one thing can wipe out the entire population. So bacteria, they have to increase their genetic variability. They can pick up new genes from other organisms or from their environment or from viruses. So we're going to look at these different mechanisms that bacteria can use to increase genetic variability. We're also going to talk about how we get antibiotic resistant strains of bacteria, like MRSA, for example. So in bacteria, the DNA can go through a process called recombination. So genetic recombination, it's just a process where organisms acquire and express genes that were originally found in a different organism. So this is how bacteria cells, they can pick up genes or DNA from other species of bacteria, and then they can actually express these genes. And there's 3 means for genetic recombination in bacteria. So we're going to go through conjugation, there's transformation, and then transduction. Those are the 3 recombination events that can occur. So we're going to look at conjugation first. Conjugation occurs between 2 different cells. And this is where those 2 cells are going to transfer a piece of DNA called a plasmid, or they can transfer a chromosome fragment from a donor cell to a recipient cell. And they do this using a direct connection called a sex pilus. So in the image over on the right, you have a blue bacteria cell and a green bacteria cell. Those 2 cells, they create this direct connection, so in between them you see this kind of protein channel called the sex pilus. And this sex pilus, it brings the 2 cells closer together. So we have our donor cell and our recipient cell. Once the 2 cells are close together, so now we're at the bottom of the image, so we have our blue cell and the green cell, we have this little channel. The red little plasmid in the top cell can actually travel through that sex pilus into the green cell at the bottom. So again, this is called conjugation. So you can have different types of cells or different species, so 2 different species right next to each other, and they can swap genetic information between them. One type of conjugation is called high-frequency recombination. This is when the donor's fertility plasmid has been integrated into the bacteria chromosome. So instead of a plasmid being passed through the sex pilus, you can actually pass a bigger part of the actual chromosome from the donor cell to the recipient cell. And then that little piece that we're interested in gets incorporated into. So high-frequency recombination, you're transferring a bigger part of the chromosome from one cell to the other. In just normal conjugation, you're just transferring a little piece of DNA called a plasmid from your donor cell into your recipient cell. Another type of recombination besides conjugation is called transformation. Transformation, this is just where your bacteria cell picks up DNA from its environment. So chromosome fragments from a lyse cell, so a cell that was lysed open or killed, these chromosome fragments are just laying around in the environment. They are accepted by a recipient cell, they're just brought into a recipient cell. And that little piece of DNA, that genetic code of that DNA fragment, is brought into the recipient cell, and it can be incorporated into the recipient's chromosome. So in the picture here, we have a little piece of DNA that's outside of the cell at the top. The cell grabs onto that little piece of DNA, brings it into the cell, and then it can incorporate it into its own chromosome. The third type of recombination is called transduction. This is where a virus called a bacteriophage serves as a carrier for the DNA from the donor cell to the recipient cell. So in this case we have two living cells again. This bacteriophage, this virus, is the one that's going to carry the DNA from one cell to the other. And there's two types of transduction. There's generalized transduction, where just random fragments of disintegrated host DNA are packed up into the bacteriophage during assembly. So you might want to go back to chapter 6 and kind of look at how bacteriophages reproduce. So how those viruses make more viruses. So generalized transduction, any random fragment from the host is put into the virus during assembly, and then that gene is inserted into the recipient cell. So any gene can be transmitted using this method. And specialized transduction, this is where instead of just random fragments of DNA are packed up into the virus, specific parts of the host DNA are incorporated into the virus, and then it's transferred from the recipient cell, or from the donor cell to the recipient cell. So here's kind of this idea. So on the left-hand side we have generalized transduction. So you have a virus that infects a bacteria cell up at the top. That viral genetic material hijacks the host cell to make more viruses. So sometimes instead of viral DNA being put into the virus, you have bacteria DNA put into the virus. So you have this incorporation of the host DNA by accident into the virus. Those viruses lysis out of your bacteria cell, and they go on to infect other bacteria cells. So now you have a green one at the bottom. If the viral DNA gets injected into the bacteria cell, the viral DNA takes over, goes through the virus life cycle. If the virus with the host DNA gets into the recipient cell, that host DNA can be incorporated into the recipient. So generalized transduction, again, any of the host DNA can be passed from one cell to another using this virus transport method. On the right, the specialized transduction, a specific part of the host DNA gets put into the virus. And then all the viruses contain this special section of the DNA. They inject it into the recipient cell. So again, generalized is basically any DNA can be carried from one cell to another. In the specialized transduction, a specific part of the host cell DNA gets transferred from one cell to the other. Besides those three different ways, recombination events, so conjugation, transduction, and transformation, you can also have something called transposons. These are special DNA fragments that have the capability of moving from one location in the genome to another. They're also called jumping genes. And they cause rearrangement of the genetic material. These jumping genes can move from one chromosome site to another, or they can go from a chromosome to a plasmid, or a plasmid to a chromosome. And these jumping genes, they can cause beneficial changes or harmful changes. So if we look at the picture on the right, up at the top in the cell we have a mostly blue chromosome with a little pink part. This pink part of the chromosome is our jumping gene, our transposon. So this jumping gene, it can move from one part of the chromosome to another, which is shown in image number 2. So it moves from the top of the chromosome to the bottom of the chromosome. The little jumping gene, it can replicate itself and insert itself in multiple regions, which is the cell in the middle, labeled with a 3. So now our little pink chromosome has inserted itself into multiple parts of the chromosome. And then in the fourth picture, our little pink piece of DNA has actually jumped off of the big chromosome into a plasmid. So you have this extra little plasmid in cell number 4, shown here. So these jumping genes are kind of unpredictable. They can move from one part to another or go into a plasmid. They can just jump around very easily. And then the genetics of animal viruses. So viral genome, remember viral genomes are made up of either DNA or RNA. And this genome, they're very small. They contain only the genes needed to produce new viruses to hijack the host cell. So this viral genome, it requires access to the host cell genetics and metabolic machinery. And once it hijacks the genetics and metabolic machinery, that viral DNA or RNA, it's going to instruct the host cell to synthesize new viral particles.